It's my pleasure to have you here officially. And I'm going to introduce you to our faculty members and researchers and students in Korean. Because you know, I'm not fluent in English, you know, so please understand me, okay? I just, you know, use Korean language a little bit.
Oh, yeah. So,
오늘 지금 이제 시간이 거의 다 돼가지고요. 아마 들어오시는 대로 또 저긴 할 텐데 먼저 좀 얘기를 좀 하겠습니다. 오늘이 아마 이제 올해 들어서 7번째 교수 워크샵이고요. 권호범 원장님하고 그다음에 박영석 부원장님이 아마 교육부 회의 때문에 참석을 못 하셔가지고 제가 부득이하게 진행을 맡게 되어서 좀 양해의 말씀을 드립니다. 한 달 전쯤에 박영석 부원장님이 제가 포스닥 때 모시던 미국 교수님을 연자로 좀 초청하고 싶다고 해서 초청을 좀 드렸고요. 아까 이제 나이를 좀 물어봤는데 계속 뭐 그냥 얘기 안 하시는데 제가 알기로는 한국 나이로 한 84세 정도 되는 걸로 알고 있습니다. 그래서 그럼에도 불구하고 아까 잠깐 들으셨겠지만은 뭐 연구원과 학생들 해가지고 지금 아주 열심히 연구를 하고 계시고 또 직접 또 실험도 하고 계시고요. 또 논문도 많이 쓰시고 뭐 과제도 지금 계속 하시고 대충 한국 포스닥한테 얘기 들었을 때는 좀 과제를 많이 쓰시는데 요즘은 좀 많이 떨어지신다고는 합니다. 근데 어쨌든 항상 뵐 때마다 좀 저를 좀 부끄럽게 만드는 분입니다. 그래서 그동안 연구를 해오면서 그 우리 보스가 느낀 점을 좀 같이 공유하면은 좀 연구하다가 이제 조금 뭐 이렇게 좀 지치거나 좀 그런 분들한테 좀 도움이 되지 않을까 생각해가지고요. 주제를 어떤 연구를 했는지 이걸 좀 부탁드리지 않고 뭐 제목에서 보듯이 그동안 살아온 얘기를 좀 해달라고 말씀을 드렸습니다. 그래서 혹시나 해가지고 미리 강의 파일을 좀 받아봤는데요. 제가 뭐 저희 미국 보스 스티브에 대해서 CV를 굳이 소개할 필요는 없겠더라고요. 그래서 직접 들으시고 확인을 하시고요. 좀 유익한 시간이 되시면 좋겠습니다. Steve, are you there?
Yeah.
Yeah, okay. So please introduce yourself to us just briefly and give us your story titled by A Lifetime of Research from Geology to the Life Science. Please, Steve.
Okay. Thanks for inviting me. This is sort of a different kind of talk that I've given, and I've left quite a bit of science in here, and I hope you'll understand. But anyways, some of this is going to be personal, and then I'm gonna try to tell a story about how I got from where I started to where we are now. Okay, and let me just start off with some general slides. There are lots of ways of becoming an academic, and I've listed on the first slide the direct route where you get a bachelor's and a master's and a PhD degree, do a postdoc, and you're a faculty member. And that is sort of the way I went. But there are other ways, including this same sort of sequence, except a lot of people will take a job in industry or government, etc., before they do academics. And actually, I did this. I worked for the National Research Council before I took my first faculty position. So these are a couple of ways of becoming a faculty member. And the other thing, by way of introduction, I want to mention to you is that it sort of is the question of how do you choose what you're going to do as a faculty member? Because if you don't choose correctly, you're not going to get tenure and you're going to get fired, or you should be fired. And what a lot of people do is they expand on their PhD or postdoctoral studies, and they expand from there. They may go in different directions, but there's usually an expansion, or sometimes you just go into all sorts of things. Whatever you did in your PhD or as a postdoc, you just begin your faculty career based on a hypothesis, and away you go, and you may go in many different directions. Now, both ways are fine. And I'll give you an example of two people that went both ways. The first one is Jennifer Dudna, who had the Nobel Prize in 2020 for CRISPR-Cas9. Jennifer started with her PhD and her postdoc on RNA, and she continued, and obviously is an enormous success. So she's an example of a person who really stuck to her PhD and postdoc training and expanded on it. Another example was Wally Gilbert, who was, again, a Nobel Prize winner quite some time ago, got his bachelor's degree in chemistry and physics, did a PhD in physics, was hired at Harvard as a faculty member in physics. And this is at the time when Jim Watson was there, and he got interested in biophysics and biochemistry, and he actually changed his faculty position from physics to biological sciences. And so he transitioned from physics to biochemistry, did a whole bunch of different things, including the Gilbert-Maxim sequencing of DNA. He founded many companies, and as a retired scientist, he's now a digital photographer. So this is an example of a person that did all sorts of different things. And I guess I've done the same thing, of course, not with the same success. I really have had an unfocused approach. Whatever seems interesting, and if there are colleagues around that are involved, I'll, if it's a little bit related to what I'm doing, I'll get into it. And my training, as you'll see, was not in what we do, but my training was in chemistry, and we, do a lot of toxicology at the chemical and molecular level, endocrinology, oncology. We're involved in drug development and interested in the microbiome and how it affects gut and distal organ health. So there are advantages to this in that you ask different questions that other people might not ask. The disadvantages is that your background in all these areas is somewhat weak, at least from my point of view, and you try to pick it up as you go. Well, just a little bit of background. I'm, from a middle class and from a small town, maybe upper middle class in the small town. No university graduates in the family. They preferred to, get out and make money. I was really fortunate to go to a private school for high school, and I really learned a lot there. I learned how to be independent. I was away from home. I lived in school, and I really got good study habits. We had to work every night, and now if I go home, well, now I go home at 7, but prior to coming here, I used to work every night, and it was just, that's the way it happened. I went to Queen's University in Canada for a bachelor's and master's degree, and went to Oxford University for my PhD. And I'll just sort of show you how and where I got from where we started to where we are now. Okay, first of all, I was enrolled in honors geology at Queen's University. Don't ask me why. I just sort of liked it. I like rocks. And then I got a summer job in a uranium mine in northern Canada. And the first day that I was to go down in the mine, there was an emergency, and they brought someone up who blew his fingers off because mining involves a lot of explosives. And I thought that it was going to be a good summer job, but that geology probably wasn't for me. And I switched to chemistry and did a bachelor and master's degree. And as a summer job, I met my wife in the lab. And that's actually was a really important time for me because I worked with a guy in the summer and for a master's degree called Bob Moyer. And again, I met my wife in the lab, and we married just after we graduated. And obviously she's been critical, but he was also really important. We both worked for him. He was not a very productive scientist, but his IQ was well above Einstein, and he was probably one of the smartest guys, if not the most intelligent guy in Canada. He was a very brilliant man, but in terms of research, he didn't produce that much, and I don't understand that, but all of us that knew him understood who he was and what he was, and he was also a very nice person. And I was really lucky in his lab. I went in early one day, And I was supposed to nitrate an aromatic compound. And instead of using concentrated nitric acid, I used fuming nitric acid. And that made a double nitro compound. And it was totally unexpected. It had never been made before and resulted in my first paper quite some time ago in the Canadian Journal of Chemistry. And that was a fun time for me. And that was important because of that publication and one other that I had as an undergraduate and a graduate student, a master's student, I got a Commonwealth scholarship at the University of Oxford in England. And that was another lucky break. Obviously, it's a great university. I don't know what it was ranked in, but it's #1 in the world now, and it was pretty good then. And I worked for a really, really wonderful guy, Prof. Jones, or Sir Ewart Jones. He was a really important chemist in England, but he was just very kind, and his wife was kind, and it was just a wonderful experience with him. And he was a great role model. My PhD work was on polyacetylenes from dahlias. And you might think, well, what the heck are you working on that for? Well, that's what he was interested in. And of course, I had a lot of fun doing it. I used to take one of the buses up to the nurseries and harvest all their dahlia stalks and flowers and take them back to the lab and dry them out and extract them and get out these polyacetylenic compounds, which the plants made, at least in those days, we thought for no reason, but they're protective molecules. They protect the plants from insects, etc. We didn't know that at the time. And that was really a fun time. And when I completed that work, I wanted to do a postdoc, and he said, well, where do you want to go? So I wanted to go to Harvard and work for, somebody famous. And he said, okay. Got on the phone, and 2 days later, I had a postdoc. I didn't have to apply for it. He just got it because he knew everybody. And the postdoc was probably my biggest failure in terms of science. I worked with Conrad Bloch, who's a Nobel Prize winner for the biosynthesis of cholesterol. And Dr. Bloch gave me a couple projects to do. The one I chose couldn't be done, but we didn't know it at the time. It was actually done about 20 years after I was there because you needed molecular biology techniques. And so it wasn't very successful, but my biggest thrill, I guess, or not thrill, but my biggest use in the lab was Dr. Bloch liked tennis, and he had a tennis court in his community. And so we played a lot of tennis. And I can remember one match where, sorry, Bloch is spelled wrong, where I played with him and Lipscomb and von During. And I think I played with Bloch and we won, but Bloch, Lipscomb, and von During were all members of the National Academy, and Bloch and Lipscomb were both Nobel Prize winners. So that was my biggest tennis match. And I wanted to not go to university right away. I wanted to get a research position at a government lab, and that was arranged to gain through my mentor in England, Prof. Jones. And I was hired as a research officer at the National Research Council in Canada in Halifax, which is right on the East Coast. It was an interesting place. We had a great lab. And I was assigned to work on a bunch of compounds that have this unusual disulfide bridge. And they're really interesting. They're antivirals. But I really wanted to also get my own independent research going. And so started some independent research along with the sort of group research that I was supposed to be involved in. And at that time, another guy called Otto Hutzinger also was a newly hired, he came a year before I did. He was a really interesting guy. He grew up in Austria during the war, and almost got bombed quite a few times by our side. And he got his PhD in Saskatchewan, and he ended up there. And he was also a little bit dissatisfied. And the two of us wanted to do our own thing. And our group leaders were not too happy about it and really didn't like us at all. And so I started out in environmental science basically because of Otto. Otto was talking to a friend of his called Vlado Zitko, and Vlado said that, you know, I think there's a big environmental problem with organochlorine compounds because we're finding them in the fish. And it looks like a lot of them are PCBs, polychlorinated biphenyls. And so Otto says, sounds like a good project. I'll get Steve to help. And so the next day he came in and said, you know, would you like to help me in this project? And I told him, no, I'm not interested. And he said, you can do the spectroscopy. It will be easy. And then I said, okay, but it better be fun. So we started on 10 or 15 years of collaboration, only 5 of which were in Halifax. And it really started me on an independent career where he did his one thing and I did another thing. But it started off, we did it together. And we were interested in chlorinated or halogenated aromatics, dibenzodioxins, furans, PCBs, and many others. And we published papers on all of them. We decided that we wanted to do everything you could do in environmental science on these chemicals, including chemistry, photolysis, metabolism, and risk assessment. And basically, that's what I did for that probably early part of my, about 20 years of my early scientific career. And this is an example of polychlorinated biphenyls. There are 209 of them. And there weren't any standards for finding out which one's which. And so one of the things we thought that was important is that if you're going to do risk assessment on a mixture of compounds, you got to know what each compound is. Therefore, you need a standard. And then you've got to know what their toxicity is. And then you can sort of add it all up together. So we started out in my lab synthesizing all 209 and reported that in 1985. And that was it's a landmark paper, probably I think it's the most highly cited paper in environmental science and technology. And so that was fun. We did that. And then we wanted to find out, and Otto was pushing this mainly, are these things that are in the environment, do they stay forever or are they broken down? People assumed they would never break down because they're stable industrial solvents. And what we showed in a paper in Nature and the Journal of Agriculture and Food Chemistry is that under sunlight, they're degraded and they lose chlorine. And the more chlorines they lose, the more degradable they are. In addition, in a paper in Science and another paper in Nature that we published, we found that these compounds individually and in mixtures are metabolized basically to hydroxylated metabolites and dechlorinated metabolites. So they're also broken down metabolically. And that at the time, which was many years ago, was fairly important, and they were published in the results were published in important journals. Now, here's the thing that bothered us and bothered me in particular, not so much Otto, this is sort of my interest getting off from him, is that all these compounds are in the environment, in fish, in us as mixtures. And one of the ways you could sort of get a biological measure of the mixture is to do a bioassay. Another way to do it is to know what the individual compounds are, know their potency, and calculate their toxicity, okay? And so that really drove me for about 10 or 15 years in terms of this field of organochlorines. And what people were mainly interested in were organochlorines that were like dioxin. Everybody's heard of dioxin. They're like dioxin. They activate the AH receptor and they cause a whole spectrum of activities. The compounds that are dioxin-like do the same thing, but they're less potent. You need more of them. And so what we wanted to do was find out how potent these compounds were relative to dioxin. And then if you knew that and knew their concentrations, you could find out the toxicity, the dioxin-like toxicity of a mixture. And that was really what drove me for many, many years. So TCDD-like compounds, PCBs, have a common mechanism. They bind the receptor and there's a correlation between their activity and their binding to that receptor. And what you do, the equation that was developed was, the first thing we did is we wanted to find out the relative potency compared to TCDD. So that's really important. TCDD is given a potency factor of 1. Everything else is a fraction of 1. And there's a variability, but methods were developed to assign a single toxic equivalency factor, TEF, for each halogenated dioxin-like compound. And that was a big effort carried on in my lab, but in many labs all around the world. And so what you could do is the toxic equivalence of a mixture is basically, and there's a mistake here, is basically the potency of a compound times its concentration. So if you had a compound that wasn't very potent, but you had lots of it, then you could work that equation, and TEQs mean the dioxin equivalence. To do this, you need to have assign these fractional toxicities, which is what we did and many others. This is mainly a WHO-sponsored thing that we've been doing for years. And so these are the relative potencies of various substituted dioxins and furans relative to TCDD. There's TCDD here, 1.0. This is a pentachloro compound, 1.0, and these are only a 10th, 0.1, a 10th is active. So the potency times the amount will give you the dioxin equivalence. And so that was, at the time, a pretty important discovery. And what's happened is that regulatory agencies worldwide have adopted this approach for risk assessment. Mixture toxicities are estimated using the TEFs and the concentrations. And what happened is that where there were problems in the world, regulations were, there were regulations promulgated. And what's happened is that we have about a 90 to 95% decrease of these toxic equivalents in the environment and in us and in our food over the last, say, 30, 40 years. They're not all gone. They're still there because they're only slowly degrading. But because we've stopped emissions based on our approaches, then our exposures are low. So that was actually a fairly important thing that we did in the early age, in my early career, in collaboration with groups all around the world. Okay, at that time, after about, I don't know, 20 years of doing this, there's only so much structure activity you can do, and basically that's what we were doing. You gotta, in order to get funded, you gotta have something a little bit more original. And one thing that really interested, so it was time to change. I didn't change that much because we were still interested in the AH receptor, which bound dioxin-like molecules. But what we were interested in was the AH receptor and its really interesting response in the fact that AH receptor crosstalk with the estrogen receptor signaling pathway, where the estrogen receptor binds as a homodimer to an estrogen response element. And this inhibitory crosstalk shows that TCDD, even though it's a bad compound, inhibits mammary tumor growth, inhibits uterine and endometrial responses, and inhibits estrogen-dependent breast cancer cell responses. So this looked pretty interesting, and this is an NIH fundable project. And so for a number of years, we worked on this crosstalk, and it turned out that in order to understand how two pathways signal, you have to understand each pathway. And we understood the AH receptor pathway a little bit, but we wanted to look at the estrogen receptor pathway, which you would expect there'd be a lot of data on. But we found that there was not much data. And so basically switching from molecular toxicology, we got into an endocrinology project on the estrogen receptor because we wanted to know how it signaled and how it signaled particular genes in a breast cancer context. So let me just here's what we wanted to do. And I'm sorry that this looks complicated, but here's a gene regulated by estrogen. When you add the receptor and the ligand, you get this completed system in which the gene is turned on. What we wanted to understand is not only how you go this way, but how you reverse it with TCDD through the AH receptor. So that was, that's an exciting project, and it's still interesting. We don't do much of this anymore. Okay, so we wanted to identify how dioxin and its receptor turned off the sequence and how the sequence was turned on. So it's a matter of gene expression. Estrogen enhances genes that make tumors grow, and we wanted to understand that. Well, to make a long story short, what we found and what's pretty highly cited now in the literature is we found that the estrogen receptor did not bind this estrogen response element, but for many genes that are listed here, cathepsin D, PS2, retinoid acid receptor, and many other CFOS, all sorts of bad genes are regulated in breast cancer, not by estrogen receptor binding to DNA, but binding to SP1 or maybe other SPs, which bind GC-rich sequences. So the estrogen, we discovered that the estrogen receptor was acting as a ligand-activated cofactor, enhancing SP-dependent gene expression. And this was very novel when we found it, but subsequently, many others have shown that nuclear receptors interact with SP to transduce gene expression. And so we published a number of papers on this, and it was a really fun time in the lab because it was we were involved in endocrinology, pure endocrinology, and some of these we were the first to discover this, and it's been highly cited. The other part of that project was how on earth did dioxin turn it off through the AH receptor? And I won't go into this in detail, but basically there are two mechanisms. One of which the ligand binds to the receptor and it goes to inhibitory response elements in an estrogen responsive gene to turn it off. The other pathway is that when you ligand the AH receptor, it binds the estrogen receptor and induces degradation. So that there's a lot of work involved in this. And we're still actually looking at this from a drug perspective now, but we don't have time to talk about it. So this was interesting. We not only showed how estrogen turns on genes, but we showed crosstalk with the AH receptor turning off genes. And that got us into endocrinology. And in addition, I got involved in a controversy. I'm still involved a little bit about endocrine disruptors and human health. And I don't know, it's an ongoing controversy. Initially, it was observed that alligators had shortened penises, so they were demasculinized. Women had increased breast cancer and sperm counts were going down. And all of these were used to say that compounds in the environment, industrial compounds like PCBs maybe, are causing all these bad things. And it's since been expanded to include almost every disease conceivable. And so we worked quite a bit on this in different aspects, but my underlying theme, I guess we published 20 or 30 papers, is that I never agreed with the endocrine disruptor hypothesis. And I actually still don't. And this may be quite unpopular in many circles. And I didn't, because a lot of people not only agree with it, but they think it's the most important thing. And I just don't. I just have never convinced myself that it's that important, but I could be wrong. That continues. Well, I just want to switch a little bit. When we were looking at ERSP1 and binding, we got interested in SP transcription factors. And let me tell you why. I'm interested in drug development. This didn't separate. And this is the SP transcription factors. There are a whole bunch of them, and they bind GC-rich sequences. And they're really important in embryonic development. But as you get older, like Joe, as you get older, these SP transcription factors go down in your tissue. But in tumors, they're high. And so that's perfect. That's a perfect drug target. And SP transcription factors are very pro-oncogenic. And so we got really interested in them, and I'm only going to show you one slide on this. We got really interested in them and are still doing a little bit of work on it. A lot of anti-cancer agents and plants and some drugs are, in fact, their activity is through downregulation of SP transcription factors. We've studied curcumin, celastrol, betulinic acid, a lot of NSAIDs, and we have a lead compound shown there. It's a triterpenoid derived from the active triterpenoid in licorice, and we've modified it slightly and have patented it and are working to try and get some financial, working with another company to try and get it. And it's a terrific compound for targeting SP transcription factors. And that's, we're still working on this, and we've actually got a grant submitted now with a company. The mechanism is complex, and I don't think we'll go through it. So let's just leave that one for now. After looking at the age receptor for a long time, it turns out that the age receptor initially was identified as a receptor that binds toxic bad compounds. But subsequently, it's shown that the age receptor binds all sorts of molecules, many of which are health protecting. How's my time? Yeah, many of which are health protecting. And so we're interested in these indole compounds. This is indole 3-carbinol. This is DIM, and they're high levels in cruciferous vegetables. They're both age receptor ligands. And so that was, we studied these for a number of years and then did an experiment which really changed what we do now. This is DIM, and let me, and it's active in a breast cancer model at about 5 milligrams per kilogram per day. And then what we did is put a big group put a big group here like this, thinking that we'd lose the age receptor activity, which we did. We call them CDIMs. They didn't activate the age receptor, but they're even more active as anti-cancer agents, 5 times more active in a rat model for breast cancer. Okay, so what do? Well, what you do is you screen receptors that bind small molecular weight molecules. And so the first target we hit was the PPAR gamma. So we screened a number of receptors. This is PPAR gamma. We activated PPAR gamma with it. One of its endogenous ligands, prostaglandin J2, and one of our synthetic compounds. So we showed that some of these CDIM compounds are PPAR gamma ligands. Published a lot of stuff on it, but found basically, and you may have done some of this, Joe, in the beginning, found basically that although the compounds were really active, they were not working through PPAR gamma. And so PPAR gamma is important. It's, all the anti-diabetic drug, many of the anti-diabetic drugs are PPAR gamma ligands, but in terms of cancer, it's not particularly important. And in addition, we had a lot of molecules, particularly this one where the X group was hydroxyl. They were really potent, but they did not activate PPAR gamma. And so what did we do? Well, we screened a whole bunch of receptors that we thought might be important in cancer. And they're orphan receptors. We screened a number and we got a hit. This is a screening assay. We got a hit, and it's an orphan receptor called NR4A1, where our compound with the hydroxyphenyl group decreased transactivation in the gene expression assay. And so what that means is that we have, and we now know that these compounds actually bind NR4A1, and they bind NR4A1 as ligands and act as antagonists. They decrease gene expression. Okay, so this is really what one of the main projects in the lab is NR4A1. It's overexpressed in multiple tumors. It's a negative prognostic factor, and it's a very pro-oncogenic gene. So it's a bad gene in solid tumors, not in blood-derived tumors, but only in solid tumors. And we published a number of papers on this. The mechanisms are similar. NR4A1 activates a whole bunch of pro-oncogenic pathways and genes, and our CDIM molecules turn it off as antagonists. And this just shows you, I don't want to spend any time on this, but NR4A1 regulates mTOR, regulates cell growth and survival, invasion. Our compounds turn it off. They're antagonists. So that's really interesting. And we're developing these and have support for these in terms of an industrial support. And we also found, this is sort of interesting, it harkens back to the ERST story, is that a lot of these genes are regulated by NR4A1 ST binding to a GC-rich sequence. And so again, NR4A1, like ER, is a cofactor that's affected by the drug. You knock out the cofactor, the gene goes down. That wasn't known before. A lot of these genes were characterized not knowing this was an important cofactor. And many of these survivin, EGFR, integrins, they're all NR4A1 acts as a cofactor through, and these are all ST regulated genes. Okay, I want to tell you, just end with this short story, which I'll finish before the hour. One of the, we've looked at 6, 7, 8, 9 cancers, but one of them that I really got interested in was a cancer called Rhabdomyosarcoma. It's a common soft tissue sarcoma in children. It's an important one, not a huge number each year. It's a muscle-derived tumor. Nobody knows why it hits kids. And I got interested in it because I was given a talk at the University of Illinois, and a guy came up to me afterwards and said, your drugs will work on rhabdomyosarcoma. And I said, well, you know, I don't know. And he kept pushing me, and he pushed me because his niece, whose name is Savannah, had a serious form of rhabdomyosarcoma. And I'll show you what she had. There are 2 types, embryonal, which is treatable, and alveolar, ARMS. These can be distinguished. And this is not very treatable. And if you diagnose it metastasis, the survival is 10%. Savannah had ARMS, and she passed away a few years ago. She didn't make it. And this is what it looks like. It's horrible. And this is also horrible. The treatment is either surgery, of course, a little bit of radiation, and these cytotoxic drugs. Just anything you can think of that's cytotoxic, they throw it at these kids. And this is important because at 45 years of age, the estimated cumulative presence of any chronic health condition in these kids treated with cytotoxic drug is 95.5%. So almost every kid that was treated with these cytotoxic drugs, either for leukemia or rhabdomyosarcoma, they're seriously sick when they're 45 years of age. So obviously we need new drugs for this disease. Now, NR4A1, we published about 9 papers on this. NR4A1 antagonists inhibit growth and survival of ARMS and ERMs, both types. And the aggressiveness of ARMS is due to a fusion gene of the PAX3 and the FOXO1. So the PAX3 DNA binding domain fused with the transactivation domain of FOXO1. So that's the FOXO1 PAX3 drives this tumor. And if you look here, here's our compound. This is an ARM cell. Here's PAX3 FOXO1A in the control. If we treat with our compound, it's gone. And these are the downstream genes regulated by PAX3 FOXO1A. So our CDIM, NR4A1 ligands, kill it. If you knock out NR4A1, you get the same thing. So it's really inactivating NR4A1 rather than knocking it out. And so that's really important. Another gene recently published, it's called G9A. It's a histone methyltransferase. It causes invasion of these cells, etc. And again, if we treat with our compound, it's gone. If we knock down NR4A1, it's gone. And so what we've shown, at least, and in addition, what we've shown is that this PAX3-FOXO1A and G9A are regulated by NR4A1 as a cofactor and they're both SP-regulated genes with NR4A1, a mandatory cofactor, inactivated by ligand. And this is just a chip assay to show that that's the case. So we've characterized this, and again, it's reminiscent of what we did 20 years ago with the estrogen receptor. But now it's for you know, a really important gene in Rhabdomyosarcoma. These compounds now, we've gone through many generations of these compounds. They're now being developed, or they've been licensed, and we're hoping that we can get them into clinical trials. And finally, 2 slides to go. We're doing a lot of collaboration because NR4A1 is exploding in terms of what it does and why it's a target. It's involved in colon cancer, and we're collaborating with a group at Methodist, and you know Maeen. Joe knows Maeen. He's now a faculty member at Methodist Hospital Institute, Research Institute, and Rob Chapkin. We're looking at endometriosis with a colleague who's from Korea originally at Baylor College of Medicine. We're looking at NR4A1 and Parkinson's disease with Ron Chalkins at Colorado State. We're looking at NR4A1 and 2 and glioblastoma with Sandeep Mittal at Virginia Tech. Sandeep is a neurosurgeon. Ted Abel is head of the Neuroscience Center at Iowa. We're looking at NR4A1 and 2 in learning and memory. We're also looking at one of our new NR4A1 ligands in terms of reversing cocaine addiction. We're also looking at new ligand applications on campus with colleagues. And these CDIMs are being commercialized through Systems Oncology, a small but fairly successful pharma in Scottsdale, Arizona. And so this is, you know, broadened my horizons a lot because these are, you know, disparate fields, but it looks like NR4A1 in our ligands will have a pretty good function. And I'm just hoping and praying we can get them into clinical trials. And finally, my last slide. Obviously, I did some of the work, including writing the papers and making compounds, but most of the work, of course, is done by students and postdocs and colleagues from the United States. I occasionally have a few Americans and Canadians, and many from China, India, a couple from the UK and Japan, quite a few from Korea, Peru, Colombia, and the Netherlands. And I was gonna make several slides, but the 150, you know, it wouldn't fit on 3 or 4 slides. So I just wanted to show you the countries. So I hope you don't mind me sort of doing a little bit of science at the end, but this is what we're doing now. And, you know, we're not too far from the AH receptor, which I started with, because the DIM compounds were AH receptor ligands. The CDIM compounds, where we added the big group, are not AH receptor ligands, but they're only one structural feature removed. So thank you. If you got any questions, I'd be pleased to answer them.
Thank you. Thank you, Steve. And any professors ask a question or comment?
질문해도 될까요? 들리시나요?
예.
Hello, Dr. Saif.
Hi.
I really enjoyed your talk. I mean, it's amazing because your work is done in like 20 or 30 years ago, but some of the work cannot be done in here still. So, but and I never forget your name because Dr. Saif is doing the research about environmentally hazardous materials and make it more safe for next generation. So I never forget your name anyway. And my question is, I'm faculty member in pharmacology, dental pharmacology department. and I tried to make some modified some compound, but you show some dioxin and dioxin-like compound.
Yes.
And the most amazing slide for me is you showed me the specific position for the chlorinated, and how you make it. Is it several different position, and how you make it 200 more than 200 different compounds specifically at that position. And here in Korea, I try to make it with collaborating with some other faculty members, but we don't have that technique. So you have any breakthrough tips to make a specific modification in already known the compounds or something?
You mean in terms of, well, for PCBs, we made all 209. A couple of them were still mixtures. We couldn't get them apart. But you know, what you have to do is for PCBs, you have two rings. So we have to bring the two rings together. And depending on the substituent, so we bring them together with two different molecules. And those molecules have certain substitution patterns. And that's how we did it. And we had to make a few, but usually even dioxins and furans, you bring two molecules together, and the starting materials are purchased usually from Aldrich. You need to be, if you have to make too many starting materials, then you're in trouble. So most of them are commercially available. For PCBs, I don't think we had to make many at all. We had one half and the other half, and there are ways to bring them together and condense them. And then you have to separate mixture. But bringing together, making two rings, we made them the difficult way. There are many other ways of condensing two rings now. There are quite a few different synthetic methods that are easier than what we used.
I see. It sounds like we need a little bit luck to get the right compound, right?
Well, you have to know what you're going to make. Actually, I spent many years making different substituents on the molecule, and it turns out, and putting them in different positions. It turns out that the first AH receptor antagonist we made by switching the positions and taking out a chlorine and putting in a methyl group. And it was just, I sort of had an idea it might work, and some of them worked and some of them didn't work.
I see.
But a lot of it, you can't make anything that you can't get the starting materials for.
I see.
So it's really important that you can get the starting materials.
Great, thank you.
Okay.
Yeah, I was the postdoc in his lab from 2005 and 2007, but he never told me that secret before.
I was afraid.
Okay. Any other questions or comments? 양윤철 교수님 톡시컬러지 좀 잘하시니까 질문 하나만 좀 해주시죠. 양윤철 교수님 계신가요? 안 계시네요.
한데
예.
질문을 지금 생각을 하고 있겠습니다. 먼저 다른 분 중.
제가 잠깐 질문 좀 하겠습니다. Steve, you have worked so hard in your life and have done so many research works. I really, proud of you. And actually today, my amazing slide is that, I knew you met Lona, your wife, in the lab, not the other place. Yeah. Anyway, yesterday I searched your name on Google Scholar and I looked at your H index. It looks it is 130 something, right?
Yeah.
And it seems to me it looks like your age. Can we schedule another your lecture for the day when your age reach your H index? Actually, I don't know whether I'm still alive at the time, what do you think about it?
Well, by that time, hopefully it'll be 160.
Okay. 양식 교수님 혹시
Thank you for your great lecture, Dr. Saif. And actually, you showed us the dioxin as a standard material for toxicology.
Yeah.
In your lecture. And I think, of course, there's a reason for you choosing the dioxin as a standard material. But I think that there are lots of other standard-like materials also. Could you tell us what kind of standard materials for toxicology study, except for dioxin?
Yeah, I'm not sure. I mean, we started with dioxins, and you know, a lot of that I got into it with my colleague Otto Hutzinger, and we continued doing a lot of stuff together. So dioxin is really the standard for me. We still use it as a standard, but there are many classes of compounds now that are AH receptor ligands that for some responses are as good as dioxin, but they're non-toxic. And so, you know, a lot of people use beta-naphtoflavone as their dioxin standard. And beta-naphtoflavone is actually used as I think it's used for some skin diseases. It's patented. But I've always sort of stuck with dioxin because I've still got some here. You know, and you don't need much of it. So that's good. But this whole field of AH receptor being identified as a bad receptor has totally changed, because now it binds all sorts of things, you know, in terms of what one of the things we're working on here now with my colleagues are the microbial metabolites in the gut. And when the gut makes AH receptor ligands, the bugs make AH receptor ligands from the food that it digests, you have a healthy gut, because the AH receptor is really important for gut health. And so now we're looking at AH receptor ligands that are derived from tryptophan. So it's really a field that's gone from toxicology and sort of everybody afraid of that receptor to a receptor that I think is pretty important. But the drug industry is only really slowly taking this to heart. There aren't many AH receptor drugs out there, but there are a couple that look pretty good that I think will make it.
Okay, thank you. Thank you very much.
김진영 교수님 질문 있다고 메시지 올려주셨는데요. 질문 하나만 어떤 질문인지 좀 해주시죠.
네, 알겠습니다. 네. Dr. Saif, thank you for sharing your interest in your pathway and chemistry. I'm a junior faculty here. Because you have been spending so much extensive time in your research, just wondering, in your personal experience, how do you manage your life and make a balance between work, family, and, you know, other related sections? Yeah. I would be thrilled to have your points.
Yeah, that's a good point. Early on, when I started, you know, I hopefully it's obvious to you that I really like what we do. I get bored sometimes, but I really like science and fooling around, and to me it's all fun. But I really, you know, I should have been home more when our daughters were growing up, but I was really lucky, and I've got a great wife. So she covered for me a lot, and she still covers for me a lot. But you know, obviously, you're as a junior faculty member, if I have people here and they have kids, and there's a choice between coming in the lab and taking care of your kids, it's much more important to take care of your kids and your wife. But I mean, I was lucky because my wife covered for me, and she sort of worked in the lab for many years. She doesn't now so much. She does a little bit. But you know, my balance was not so good. And and I think I would recommend someone to be more balanced and be more focused when you're at work. Sometimes when I'm at work, I play games on my computer and whatever. I'm not as focused as I should be. But I think, you know, you can do great science and have a balanced life. But, you know, you still have to work. You have to get in the morning. You don't take off days, and you got to work some on the weekends. But you can still lead a balanced life as a productive scientist.
Okay, thank you. I got your points, Dr. Saif. Thank you.
Yeah.
When I hear the Dr. Saif, your voice, I think it's time to bed for you, right?
Yeah, almost.
Okay, so we've just closed your seminar right now, and thank you for your great talk, and see you
Okay.
soon, in Korea.
Yeah. Okay. Thank you.
Zoom seminar.
Bye.
Okay.
Thank you.
오늘
Bye.
Thank
오늘
you.
참석해 주셔서 너무 감사합니다. 이상으로 워크숍을 마치도록 하겠습니다.